Welcome back, everyone. Lunch was good? Yeah, right? Okay, we are going to continue today, and it is my pleasure to introduce my colleague. He is a biostatistician and assistant professor here on campus, and we are both part of the biostatistics department, and he is with the CDA group, the consulting biostatistics group that we were talking about before. And he is an expert in adaptive trials, and he's a local expert, which is really exciting. And he is a lot of fun to work with. So without further ado, let's welcome Alex. Thank you.
Thank you, Liz, for that introduction. And today I'm going to be talking, as Liz alluded to, this idea of adaptive clinical trials. And I titled my talk somewhat tongue-in-cheek, from basics to Bayesian, because I was going for the alliteration approach. But I will note, just because things aren't Bayesian doesn't mean they're basic, and vice versa. But I think the approach today we're going to take is to look at a really high-level overview of adaptive trial designs. Each one of the topics I'll be introducing and talking about could in and of themselves serve as an entire 1 hour sort of talk or presentation or seminar. And so I think what might be most helpful for those of us in the audience is to get a taste for a lot of different elements of a trial that could be considered adaptive, or things that we might be able to work towards as adaptive elements of a trial. And so I'm going to start just by introducing broadly what are adaptive designs. We'll then walk through some of the specific elements and some examples of them in either practice or how they might be implemented in a high-level situation. And then we'll also touch at the end on the idea of Bayesian methodology, motivated for adaptive trial design, but really more of a general consideration if you're trying to look at using Bayesian methods in your next trial or design. So what are adaptive designs? This is a question I get asked a lot as I'm meeting with collaborators or people who are interested in trying to explore these topics for their own research. And so because it's a very popular area right now of research, I think one of the first sort of responses I get is that it's the key to all of my research woes. I've found it, I've solved it, I'm ready to go. This adaptive design will fix every problem I had before. Alex, when you said that I needed a sample size of 200, let's make it adaptive. Make it 50. And then I just politely nod along for a moment as I let the gravity of the situation sink in. So maybe they have some potential to cure some of our research woes, but there might be some more nuance. So what's another way people might think about adaptive designs, or at least that I've encountered in my experience? Well, maybe they're designs where I can do whatever I want, whenever I want, to ethically answer my research question. I mean, they're adaptive after all. What am I adapting if not whatever I darned well please throughout the trial? Again, we'll see how this is not quite at least meshing up with the FDA definition of an adaptive trial design, but we can note that there is some potential for flexibility, just maybe not unlimited adaptability. There's also this thought when I do tell people about adaptive designs that maybe these are the good designs that I've been keeping to myself and selfishly not sharing with collaborators. Again, they are, in certain circumstances and contexts, fantastic designs and opportunities to use and utilize these elements. There's also chances, though, that they're not appropriate for your problem at this time or this specific context you're trying to use. So I'm not just selfishly keeping them to myself. I am actually actively trying to help you design your best study to answer your research question. So let's see, what does the FDA actually define an adaptive design to be? And so it's cut off a little bit in the bottom here for our citation, but we see their definition from their 2018 guidance document from last fall is an adaptive design is it defined as a clinical trial design that allows for prospectively planned modifications to one or more aspects of the design based on accumulating data from subjects in the trial. And so hopefully you'll note that I'm calling out some attention here to the underlying prospectively planned. We're not doing whatever we want, whenever we want with an adaptive design. We are actually planning these modifications in advance. And another important thing to keep in mind as we review some of these examples that do exist for adaptive elements is that we're really doing these adaptive moments or chances in the trial based on the accumulated data we have thus far. So it's not something that we are willy-nilly just approaching. We're actually trying to inform it based on evidence that we don't yet know what will occur because it's our future trial. So there's a lot of different things we could make adaptive in a trial. And this is one of the challenging things. When people hear of adaptive designs, they think immediately, oh, I'm going to do this one thing, or it means this group of things. But in fact, there's a lot of nuance and possibility for what this could mean to each person and in your context. And so from the FDA, they've outlined really nicely in this guidance document some broad classes of elements that they think of as adaptive. The first one is actually more of a sort of traditional approach in clinical trial design, which are these group sequential designs. Or in other words, we're planning in advance some interim analyses where we might stop the trial for efficacy or futility, but we're doing something that's more well-defined throughout the statistical sort of research, literature, and history of trials. So surprisingly, when some people ask for an adaptive design, they're a bit taken aback when I mention actually just doing a group sequential design is adaptive in and of itself, because if we think back to that definition, we're planning in advance for a potential modification, and we're going to do so based on the accumulated data we have. And so in today's talk, I'm not really going to delve into different group sequential designs because there's a lot of rich literature out there already, and I think there's a lot more novel aspects that we can really tease out and talk a bit more about. One of the first things we will touch on is this idea of adaptations to your sample size. So in other words, we're going to re-estimate the sample size potentially in your trial to adjust issues such as unplanned changes or misplanned or misassumed responses in your trial during that stage when you're powering and doing a sample size calculation. There's also the potential for adaptations to your patient population. This is known as adaptive enrichment, where you try and hone in potentially on different aspects of your group or your population you're enrolling to really drill down to the question you want to answer. There's also adaptations to treatment arm selection. So this idea that we could potentially add or terminate arms as the trial's going on. So rather than starting with some set number of interventions, we can flexibly remove or incorporate new ones as the trial moves forward. Another really popular area that we'll discuss briefly is adaptations to patient allocation or adaptive randomization, where we actually can allocate people differently than we might have planned at the start of a study. Something we won't touch on today, but does exist in the guidance, is this idea of adaptations to endpoint selection, or honing in on how you'll actually evaluate your trial and your study when it's complete. And finally, the catch-all answer the FDA gives, as well as adaptations to multiple design features. So they present each one of these items as a standalone element, but we can imagine you can mix and match them in different ways that might be most appropriate for your trial in a given context. And so without further ado, we'll break down some of these topics with very brief high-level overviews of what these might look like or mean for you as you're thinking about potentially incorporating them into your next trial or study. And so we'll start by looking at sample size re-estimation, or that adaptation to the sample size. And we're going to start here with a hypothetical scenario, asking ourselves, what situation might I be interested in even considering this adaptive element for my study? Or is there a case where it might be potentially useful to look at? So let's assume you've done some research, it's in a fairly new area, and you've designed this study, and you're powering it, you're doing a sample size, but you have some uncertainty about that assumption you've made. You think, maybe things will be different actually in this trial or this study I'm designing, and I'm worried that these assumptions that I've made are incorrect and actually might derail my future success of the study. So let's say now we do our study actually, and as the study is being conducted in this hypothetical scenario, the observed treatment effect is smaller than you might have planned for. And so maybe you were a little overly optimistic, or the previous data you did have suggested something that you're not observing. But you can note that this observed effect is still clinically meaningful. So you'd still like to come to a conclusion that if I did observe this effect, it's still beneficial. It's still worth sharing that information. But the way you currently designed the study might not have an adequate sample size if that effect is smaller than you would have expected originally. So if we maintain the planned sample size, we could potentially in this scenario be underpowered to detect that difference, which means we might potentially end with a negative trial, or a trial that doesn't show the hypothesis we were trying to show in the first place, even though we do have the potential for some efficacious, clinically meaningful responses. So in this approach, with sample size re-estimation, broadly speaking, we'll generally try and look at some interim analyses or interim time points where we'll try and say, okay, We want to address our prior uncertainty by looking at the data we've accumulated so far to see if it matches with what we assumed. And if not, can we adjust the course of our trial so that we can maybe enroll additional participants or subjects in the study to address this difference or deviation from our original assumptions. These approaches have a lot of different developments that have occurred over time and can be both blinded and unblinded. And so sort of as we think of in a trial, a blinded re-estimation procedure would suggest that you actually don't know what estimate's coming from what group. And unblinded would suggest that you actually do have some information about what data is coming from which group or groups in your trial. The FDA guidance focuses specifically on unblinded contexts, but they do allude to this fact that there are other methods that have been and are being developed for blinded approaches. The FDA does recommend, as with any sort of intervention or change you might make with the trial, to really consider those changes and try and maintain the integrity of your trial. They use the example with an unblinded approach that if you have a really savvy collaborator or investigator and you give them some information about the sample size and its re-estimation, they could potentially back calculate to some conclusions that maybe weren't supposed to be public or common knowledge for your study, potentially violating that integrity of the study or trial you worked hard to design and conduct. One of the challenges, though, with this approach, so when I first introduced this method to a lot of people, they think this is great. This is a check on my assumptions to see if I'm going on the road to success, or maybe if I need to reallocate slightly. However, one of these scenarios that could happen is that we actually might need a much, much larger sample size to observe your difference that we see in the trial. In this situation, if you already have a very large trial, let's say with 10,000 people, as we'll see in the example in the next slide, in that case, adding 1,000 more people, not great, but also you're like, I already have 10,000, I guess I can achieve that. Whereas if your study size is even 50 people, and I say, oh, you need 50 more people, you need 100, you're literally doubling your study size, and in that case, you probably didn't plan for that in the first place. Otherwise, you might have allocated resources to address that concern. So as an example of a trial that actually puts this into action and tries to use this approach to address this concern about the uncertainty of their estimate, we're going to look at the, it's covered here on our bar, but the Champion Phoenix trial here was looking at the effect of platelet inhibition with cangrelor during PCI and ischemic events. And so some background of this trial and the very high-level process of what they were working for. They designed the study under some assumptions of a composite outcome of 5.1 and 3.9% in their two study arms. And so when they put these calculations into play, they realized that they would need 10,900 patients enrolled in total to achieve 85% power and a two-sided type 1 error alpha of 0.05. However, they were concerned about the uncertainty around these estimates for composite outcomes of cangrelor and its comparator treatment, and so they planned that they would do a unblinded sample size re-estimation after 70% of their participants were enrolled. So that way, if there was some difference in that 5.1 and 3.9% difference, they could try and address it at this stage while they still had some time left in the trial's duration to address that potential lack of sample size they might have. Now in practice, when they actually did this, they had their one interim analysis after 70% of people were enrolled, and they actually identified that at that point, according to some minutes and some FDA reports, they actually had an early crossing of an efficacy boundary. So they potentially actually could have stopped the trial at this point with 70% of the participants because they already demonstrated the benefit of cangrelor over the competitor. One of the things this tells us, though, is that there was no need for a sample size increase because if they actually were adequately powered enough to detect a difference at that stage, they didn't need to enroll hundreds or thousands of more patients. But we can also note in this case the sort of nuance and power of data safety monitoring boards, because in this case they didn't actually stop the trial even though efficacy was shown because they wanted to collect additional safety and secondary outcome information to use in their trial. But this just serves as an example that a large study in this case planned in advance for an intervention or an interim analysis at 70% to potentially alter the trajectory of their trial. It might be something to consider if you have uncertainty around your estimates and want to safeguard against the potential for having an underpowered study. One of the other things we can consider doing is this idea of adapting the patient population or adaptive enrichment. So again, we're going to consider another sort of toy hypothetical scenario where we might say, well, why would I want to quote unquote enrich my patient population for my study? So let's assume we have this nice hypothetical scenario where we're enrolling yellow smiley faces and purple smiley faces. And so we expect that between these two individuals, the yellow smiley faces actually have some more meaningful or strong benefit to this intervention than the purple smiley faces. And so in one approach, we might do what we see here, is that we say, we think this might be the case, but we're not really sure. So we're going to enroll everyone in the population to our trial, try and do inference on that, and maybe come up with some conclusions. An alternative scenario, though, is that you feel really strongly that you have some prior evidence or hypothesis that that yellow treatment really works better, or potentially you expect that the purple treatment group maybe doesn't work at all. Maybe it's some sort of genetic mutation where it affects the regulation of some medication in a person. So you might plan to enroll only a targeted subpopulation. And so in this case we see here, we have this alternate universe where we designed a study that only enrolls yellow smiley-faced individuals. In this case, we're only going to be able to make a conclusion on these individuals in our population, but it provides us with a very specific answer to one question. Now, there's some reasons that we can discuss about why we may want to or not want to do this sort of population enrichment, or having to choose between the dichotomy of either one of those approaches. One reason that we might want to use this approach of population enrichment, which we'll see as an example on the next page, is that we want still to get some information about both that targeted yellow smiley face subgroup, but also collect information on the purple smiley face subgroup. We think maybe we actually don't have enough evidence that it doesn't work or doesn't work as well in that group, and we want to collect more information. There also might, again, be uncertainty about that treatment effect in that population, so we just want to include them in the study so we can at least evaluate that within one trial instead of multiple trials. We can also note sort of a benefit why people are attracted to these designs is that there's the potential for greater power relative to a fixed sample design where we just enroll everyone without restricting just the yellow smiley face subgroup. We can see this, I think, on our next page where we see a very simple sort of example of how this design might work and flow through the trial. And so we see in an adaptive enrichment design, we start with stage one of enrolling both groups, so both our yellow and our purple smiley face individuals. At the second stage, we have some interim analysis where we're going to evaluate these targeted subgroups that we a priori have specified. We're going to determine if we should continue on with everyone, so going up this sort of top route to 3A. And so we have a kind of traditional trial structure here where the entire time we're enrolling everyone from both populations. Or do we want to actually restrict our enrollment to the next stage of the trial to only focus on these yellow smiley face individuals? Perhaps we've identified that there isn't promise shown in these purple smiley face subgroups, and we actually want to increase our power by focusing our second stage enrollment of the yellow smiley face individuals to have more information for those people. And so at this stage, we can imagine that we have really two potential options that, again, we decide a priori to make that change, and then follow through with some thresholds or decision rules that we had defined in advance, either enriching or maintaining the same subpopulation of interest. The next design piece that we're going to touch on briefly is this idea of adaptations to treatment arm selections, and we're going to focus right now on this idea of what are called seamless designs of a few varieties. We'll also touch a little bit later when we get to master protocols on another approach that has a lot of flexibility with this idea. So seamless study designs are this idea that in a traditional clinical trial process, we have multiple stages of development. Maybe we have our distinct preclinical and then a phase 1, a phase 2, a phase 3, potentially a phase 4 trial. And each one of those tend to be a standalone, independent trial that you've designed and conducted. Of course, this takes a lot of work and time, and so some people have proposed trying to combine multiple phases of these studies together. So for example, instead of a separate phase 1 and phase 2 trial, you do a seamless phase 1-2 trial. Similarly with a 2-3 design, where you seamlessly go from phase 2 to phase 3 without having to stop the trial, develop entirely new infrastructure, and do an entirely new design. In this process, generally, we're going to rely again on this idea at some interim analysis or analyses, we're going to make some decision about the treatment arm or arms we have included in our study. And we'll see this on our examples on the next few slides. And these decisions ultimately help us determine what continues from phase 2 potentially to phase 3, or phase 1 to phase 2, depending on the type of design we're looking at. The advantages of these designs are that we have the potential to reduce our overall study size compared to having to do standalone separate trials. We'd have shorter development time. So especially if you're working with new treatments that you want to get out to people more quickly, you're able to reduce that development because you don't have to reinvent your trial. And there's also the possibility for more long-term safety information. So you can imagine that in a phase 2 study, when you start to enroll those people, if they continue on into your phase 3 version of the trial, you already have all of that preceding time of information to take account of and also continue forward to also look at the effect of your interventions. Some of the disadvantages or trade-offs of these designs, helping to illustrate why adaptive trials aren't always ideal for every situation, include some logistical challenges, such as how will you seamlessly transition and how will you make these rules or identify when, in special circumstances, you address some limitations. And also there can be issues with maintaining certain statistical properties. So you tend to need some advanced statistical methods and working really closely with your statistician or methodologist when designing these types of studies. One version of a seamless phase 2-3 design that I really like and I think is nicely illustrated comes from this European Patients Academy on Therapeutic Innovations. And so this is just one resource I like to recommend to people if they're looking for a high-level overview of a lot of clinical trial elements and design types, because they really nicely lay things out with graphics like this and give nice examples and motivating context at a fairly high-level overview to really help get the message across to a broad population. In this case, though, we'll assume in their seamless phase 2-3 design, we have 3 potential treatment arms that we're going to enroll to and consider in phase 2. We have our red standard of therapy control arm, and then 2 treatment arms, a purple and a green arm. And we're going to assume here in their example, participants come in this box and they consent to be part of the study, and then they get randomized to one of these 3 arms where we follow them during the course of the trial for whatever we've designed. In this example from the European Patients Academy, they assume an outcome of progression-free survival, or PFS. And we can see here, if you kind of focus, I missed this the very first time I looked at this graphic, but these colors of the little pills at our interim period with this first PFS are the purple and green 2 treatment pills. And so what they're trying to illustrate here in the seamless design is that this first interim analysis, we're really trying to make and evaluate a decision about which one should we continue on to phase 3. Can we decide that one of these treatments is actually more promising or better, and then stop the enrollment of one of these other arms, dropping it from the trial, as we seamlessly transition to a phase 3 study? Some of the advantages here is that we have all the information from phase 2 and phase 3 to continue for both the red and green arms in the figure. So we have this larger sample size to draw upon, as well as potentially choosing different, in this case, the same outcomes for our phase 3 trial of efficacy. So there's a lot of flexibility with these seamless designs to consider to help bridge some of the gaps we have with more traditional approaches to trial design. Another really popular approach is this idea called multi-arm, multi-stage designs. And so we see here a kind of similar setup. We have at the left of our European Patients Academy image, 4 different novel treatment regimens we're considering, and 1 shared common control regimen. So we can imagine here one of the advantages is that we're not going to do 4 separate trials with 4 independent control arms where we have maybe just treatment 1 to control and a different trial with treatment 2 to control, all happening simultaneously. We're going to design one study that includes them all within one approach and one protocol. In this case, we have our interim analyses, and we'll have some predetermined threshold we use to drop arms that we don't think are encouraging. And so, as we'll touch on a little bit later when we briefly introduce Bayesian methods, we might have something here, if it was a Bayesian design, like the predictive probability that this novel regimen 2 might be shown to be efficacious at a later stage, and if it doesn't meet a certain criteria, we drop it, because we want to focus on enrolling people to arms we think A, might be more efficacious, and B, by focusing on those arms, we have the potential to increase our power for any final conclusions we make. And so we see here in this example they give us, by the time we get to this far right sort of threshold, it's the end of phase 2 and the beginning of phase 3. And we see in this example, we've narrowed it down to a sort of more traditional comparison now of the novel regimen 3 to the control regimen. One of the disadvantages here, at least with the way they've just presented and structured their trial, that some people might consider a disadvantage depending on your hypothesis, is that it's not designed at any point to do pairwise comparisons between your different regimens that you're of the novel regimens. So if one of your hypotheses of interest is that you really want to look at all possible interactions or comparisons, this design might not be most appropriate, or at least might not have the most efficiency relative to just utilizing a shared control arm for your comparisons in this example. So one of the final adaptive elements we're going to talk about is this idea of adaptive randomization, or adaptations to the patient allocation in the study. And so in this case, we can imagine that a lot of traditional trials, we might assume sort of a simple randomization structure of one-to-one allocation, or 50% to a control arm, 50% to an intervention arm, and we're hoping to maintain that throughout our trial. In these approaches, we're going to look at ways we can adapt that based on some different motivations. So the first type of adaptive randomization we're going to discuss is this idea of baseline or covariate adaptive randomization. This is a randomization approach that adaptively places people based on their baseline characteristics in the trial when they come to enroll. And so in this case, the probability of your treatment assignment is actually altered on the basis of who's already in the trial and their characteristics of interest. So there's some different names or approaches people have for these, such as biased coin randomization or minimization procedures for baseline adaptive randomization. But essentially our goal is to try and address some limitations we have where we might say, okay, I'm doing a study and I'm really interested in maintaining, let's say, sex between my study arms. So I'm going to stratify on sex as a variable. And then let's say somebody else says, oh, I think we should also maybe stratify by race to ensure balance of race and cross our study arms. And you're like, okay, that sounds good, 2 characteristics to balance on. And then someone says, I think we should also stratify on income distribution, broken up into, I don't know, series of 5,000. So we suddenly have 30 different income distributions. Now you're like, okay, I have like a lot of different things to stratify by on right now, and I'm not really sure I'm actually going to get people in all of these different combinations to even randomize in my study. If we were to instead treat these as a set of covariates that we're wanting to balance on for some motivating scientific reason, we might be able to actually take advantage of this baseline adaptive randomization instead of trying to stratify the heck out of our study design. And in this case, we have some considerations just to keep in mind if this sounds like something that might be of interest to you in a future study. One is, if you're doing a multi-site trial, do you have some central entity that's in control of this adaptive randomization, or is it by each specific site? If you have 2 treatments, it's fairly simple, but if you expand it to multiple treatments or maybe incorporate it into a design that can drop treatment arms, how do you incorporate this adaptive randomization and maintain the integrity of your trial? We also have to answer the question, what is a lack of balance? Like, what does that actually mean in the context of our trial? And part of the challenge here is that there's multiple approaches for these minimization procedures that use different definitions that might vary its appropriateness for your specific context. There's also really the big question of what covariates do I want or feel like I need to use to maintain balance? Because if we just start to throw everything into the design, we actually might not be balancing over meaningful covariates in the first place. And so it's something we need to thoughtfully consider before implementing this type of randomization. The big advantage, though, of this approach is that we can balance this overall composition with respect to our variables of interest without having to do a whole bunch of stratification of our randomization schedules and schemes. Another really popular and common type of adaptive randomization that people show interest in, and largely for ethical motivation, is this idea of response or outcome adaptive randomization. So now, instead of trying to balance information based on people's baseline covariates, we say, well, what if we want to actually assign more people to arms that look like they're encouraging or showing some benefit? So in this case, we'll actually look at our assignment probabilities as changing based on previous individuals' responses in the trial. The motivation here is that we want to allocate as many patients as possible to what we consider the best treatment arm possible in the study. And so if something is showing more promise for an outcome, we want to try and give more participants that treatment to try and get more information on it and also give them the chance at receiving that efficacious treatment arm. There are some challenges, though, and important limitations with this approach. Some recent research has shown that in certain circumstances, this actual design approach can backfire and inadvertently randomize more people to the quote-unquote inferior arms. There's also concerns about sample size imbalance, because you can imagine when you design your original study in advance before seeing any of these outcomes, you're probably going to make these assumptions based on like an equal allocation to your study arms. So you're powering based on one set of assumptions, but suddenly if we start randomizing more people to one arm and then less people to other arms, we might have a more challenging time to actually try and identify effects because we have an imbalance in our sample size now. And there can also be challenges where there are time effects present. So if the response changes over time, suddenly we might be randomizing people based on previous information that no longer reflects the status quo or the present time response. One sort of nice example of this actually comes from back in 1969 from Zelen, and it's this idea of a play the winner design. And so I think it's just a nice illustration just of the kind of motivation for one sort of simpler approach that doesn't necessarily rely directly on complex minimization formulas. So in this approach from Zelen, the first step is that you have one person start in your trial and you randomize them to either of your arms with equal probability and you observe how they do. Depending on what their response is, we have sort of two courses of action. If they show a positive response in their randomized arm, you say, that's interesting, it shows some benefit potentially or evidence for encouraging results in that arm. And so we might try and increase the randomization chance in the next person. The other opportunity is that we might observe that they didn't show the benefit in that arm, and so we say, oh, well, maybe the other arm will hopefully show benefit, and we should increase the chance in the next stage that that person will be randomized to that arm. And we can do this for each person who enrolls in the trial after we observe their outcome. Some of the disadvantages or challenges are that it makes sample size and power calculations, again, really challenging from the sort of statistical side of things because you actually don't know what we're going to see in advance. And we also have this potential challenge of really needing to know the individual who enrolled previously, what their outcome is, before we can update the randomization ratio or adaptively randomize the next individual for our study. However, some people have noted it is possible to overcome this by updating in blocks of individuals instead of each specific individual in your trial. And so there are ways to address this and get around it, but it is still something that can logistically make things a bit more challenging to implement or evaluate in advance. So I'd like to change gears slightly now. And so we've touched very briefly on a lot of the adaptive elements from the FDA guidance. One area, though, that these are being applied more frequently and are also a really popular area of research that I also focus on is this idea that's known as master protocol designs, or umbrellas, baskets, and platforms. And we'll see what these mean in the upcoming slides. For our motivation here, there's this really nice New England Journal of Medicine review article by Drs. Woodcock and Lavange, who look at this idea where their title really nicely summarizes what a master protocol broadly is, which is something that can study multiple therapies, multiple diseases, or potentially both of those things within a single trial design. So traditionally, when we think about a clinical trial or designing clinical trials historically, we think about them as a series of sort of standalone trials where we're trying to address one very specific question, perhaps, and we're trying to do this in a very targeted population. One of the challenges we have with this, though, is that resources are becoming increasingly constrained, especially as there's just a lot of good and interesting ideas to test. And these can be prohibitive in trying to do a bunch of standalone trials as we once used to do. Also, with this more recent sort of turn towards precision medicine, we need greater flexibility in our designs to really help identify what patients might benefit most from therapies or interventions of interest in your trial. So one way that people have addressed this is by looking at these master protocol designs, because they provide a unifying framework within a single or master protocol for a study to answer potentially multiple questions. And so instead of having to design a whole bunch of separate trials, each with their own standalone protocol that you're trying to develop and use, these designs use one single large overarching master protocol to address those questions. One of the reasons people are really turning to and liking this idea of master protocols is a lot of the innovation that's kept within. And so Woodcock and Lavange discuss this in their paper, and I'll just highlight a few things from one of their tables here. But the nifty thing here, I think, is that the innovation isn't just on the statistical side of things. It isn't just from adaptive design elements, but it also has to do with the trial infrastructure. So a lot of the practical, actual implementation components that go with having to run or conduct a study or trial. And so we see here with adaptive trial designs, we have the chance to have common screening platforms, especially if we're using some sort of biomarker within the trial or it's a biomarker motivated study. There's a chance to use similar or the same governance structures. So instead of having a ton of different data safety monitoring boards or committees or steering committees, you actually can have single entities for each one of them in your master protocol. There's also this chance now with the advent and growth of trial networks and clinical centers to cross-work across institutions and different entities and pool resources together more flexibly. You can also address some of these concerns about how do we do randomization or safety monitoring and quality control by having a single overarching protocol. Within the design pieces, there's a lot of opportunity to include a lot of these randomization or other adaptive elements we discussed, longitudinally modeling and following patients throughout the course of either disease, a treatment, or a parallel arm of a study. We see here that there's some examples here where you can, as in some of those previous designs, shared controlled patients like that multi-arm, multi-MAMS design, the natural history cohort, which you can enroll if people don't qualify for a specific arm in your study, but you still want to follow and collect information on them, or to qualify various biomarkers in a study. So again, Woodcock and Lavande are trying to also address some of the challenges we've had recently, in that there's a lot of variability in the language people use to describe these design. And when someone said umbrella or platform or basket trial, they might have meant something completely different from the person sitting next to them. And so they proposed this common unifying structure for definitions to try and give us a common framework to work from. And so very briefly, we see here that an umbrella trial is this idea where we have multiple targeted therapies, but we're focusing on one disease. A basket trial is sort of the exact opposite, where we're focusing instead on a single targeted therapy, but with multiple diseases or disease subtypes we specify. As we'll see in a few slides, a platform trial is a very flexible, very broad trial design that gives you a lot of flexibility to incorporate various elements in these and other adaptive components we've already discussed. So I think there's some nice graphics that help to illustrate what these designs really are getting at or mean. So we see for an umbrella trial, we're looking in this example at lung cancer as our sort of one type of cancer. We're going to assume that we've looked at genetic mutations for the tumors, and we think that actually different genetic mutations might be better addressed by different targeted therapies. And so in this case here, we see that we'll give the orange mutation, test drug 1, people who have a green mutation, test drug 2, and people who have the blue mutation will qualify for this test drug 3 to try and really target for that individual what might be most efficacious. For a basket trial, we see instead of the one type of cancer, we now expand to multiple sites of the cancer, but we're looking at a shared genetic mutation across those tumors in different sites. We have maybe one drug we've developed that we think will target that mutation, but we aren't sure how effective it will be across sites or if there's other considerations to take into account. And just on this slide, here's another graphic representing umbrella and basket trials from the Woodcock and Lavande paper. And so one example I want to spend just a few moments walking on, because I think it's a really flexible system that's really going to be used a lot more moving forward, is this idea of platform trials, largely because of the insane amount of flexibility you potentially have in your design. And so within this trial here from the Woodcock and Lavande paper, again in the New England Journal, we see that they start their trial and they have some period of ongoing continuous screening that just goes as our x-axis here of time, just keeps going on in sort of perpetuity, a very flexibly designed study. In this case of their example, they're trying to illustrate a case where we might be trying to allocate people based on some biomarker, so borrowing potentially from that sort of umbrella or basket trial design, depending on what our motivation might be. And we see here within then each of these stratum, we have different groups that people can be randomized within. And so for biomarker A, we see there's a standard of care and 2 potential drugs we want to consider. Biomarker B has its own standard of care and its own drug to consider. And we also have a negative biomarker group, which in some other examples actually might be a natural history cohort, where you, instead of randomizing people to treatments, you enroll them and let them follow whatever their standard of care is, on the off chance a future change in the trial might show them to be potentially eligible for a different portion of the study, such as the addition of a biomarker C stratum in the future. We see here then within their example, there's a lot of flexibility where investigational drug 1 is stopped early because it shows efficacy and it shows a benefit. And at that point, we see it also becoming later on the new or revised standard of care within this arm. We see that drug 2 is an example of where they allocate some number of individuals and they reach target enrollment. and they close recruitment at the end to eventually make their decision. We can also add new drugs as the trial is ongoing or entirely drop different stratum depending on performance. And so there's a lot of flexibility with these approaches for platform trials that we can utilize and take advantage of. So just broadly for these master protocols, these designs can be both comparative or non-comparative designs. They can be exploratory or confirmatory. They can or cannot have adaptive elements. So really there's just a lot of flexibility. There's a lot you can do with these designs in your framework as long as you're willing to put in sort of the increased burden and challenge of having to design a study with multiple parts that has some uncertainty as to how long it potentially might actually be ongoing for. So we have a few examples here that I'm going to skip over right now for the sake of time that you can read through on your own, but essentially we have an example of an umbrella trial in lung cancer. And I will just point out, just for drawing in from our earlier point, they did involve outcome adaptive randomization where they have an equal randomization period and then they adaptively allocated people where we see more people get this sorafenib treatment in their design. We also have an example of a platform trial design for Ebola in West Africa back in 2013 to 2016. And so we just have some examples there of the motivation and some of the reasons that they tried to do this trial design at the time. As well, here I provide an example from the design of what it might have looked like in a simpler context for a platform trial than the Woodcock and Labange paper. And so in this case, we're sequentially considering a series of two-arm trials where the adaptive and innovative design components come from the fact that it's a seamless sort of transition from segment 1 to 2, 2 to 3, and onward, and without having to stop each trial and design an entirely new study. There's also some extensions we can make, but I'll just mention again briefly because this is some of my own research interests for methods, is statistical approaches to incorporating supplemental or past information. And so in the case of Ebola, because there was a concern with the time effect of the underlying disease mutating and potentially becoming immune to certain treatments, they were worried about incorporating past information at future segments, which is potentially inefficient because there's a lot of data from past participants we could use. So there are statistical methods that I'll mention that do evaluate if you can incorporate this past information. And so with those pieces in place, I'd like to spend just our last bit of time briefly discussing this idea of utilizing or incorporating Bayesian methods into your trial design, or considerations we have to keep in mind when we do want to think about using these approaches. So from everyone's favorite rigorous academic source, Wikipedia, we have this nice little table summarizing Bayesian and frequentist approaches with Efron's adjectives. I think sort of the big piece, if you aren't familiar with Bayesian methodology, the big, I think, sort of double-sided sword is that you have a prior specification involved in your process. And so it can potentially be beneficial because you can incorporate meaningful information into your trial and meaningful design pieces. But it's also subjective and could potentially incorporate information that could bias your study. And so it has some potential, but also to be carefully used, which is in comparison to the frequentist method where you might assume using a certain test, and that t-test will give you the same response no matter what you do because you're not choosing maybe a different prior for different situations. One fun quote that I will mention, just in case people are like, I would never be a Bayesian, is that really everyone's a Bayesian in the design phase of your study. You're using past information, prior studies to make some assumption you're doing for a future design or future trial you're doing. So really you're incorporating previous information to inform some future aspect. And so if you're really opposed to Bayesian methods, just maybe think retrospectively a little bit and introspectively about where you might actually be inadvertently using these approaches. To define a bit more about how the FDA sees a Bayesian adaptive design and what we actually mean when we say Bayesian adaptive design, we can give this very broad definition, but it's essentially any designs that use Bayesian approaches for either the statistical reasoning and or calculations of your study, with some potential examples the FDA provides of being predictive statistical modeling. So this goes back to that potential in the MAMS design. We're trying to, in advance, at some early stage, predict if we think there's even a chance of there being efficacy in the arm, given the information we've observed so far and what we might observe possibly in the future. There's also, because it is Bayesian, using prior information. And so the FDA gives this example of where for a dose response study, you might have some previous information about how you think things might respond, or you want to try and control things from maybe going to too high of doses too quickly. And so you can put some prior information into these designs to help govern the dose escalation and selection to avoid situations where you rapidly ramp up that dose to potentially toxic levels. And again, an area of my own personal research interest is this idea of borrowing information from external sources. And so the FDA gives a very broad definition that maybe previous trials or natural history studies, maybe registries. My own research focuses mainly on comparing similar to similar things. So if you're doing a trial, trying to maintain that same level of rigor in incorporating that information. Well, there are different ways you can incorporate this stuff through other sources that maybe are not on the same level of hierarchy of information or evidence as well. And also, one of the most important things to mention is sort of the key component that might be the most direct parallel to a frequentist design that we're used to is the use of posterior probability distributions to determine the success of our trial's outcome. So this is really in comparison to maybe using a frequentist p-value for our conclusion. So instead of a p less than 0.05, we might define some or calculate a posterior probability that we've achieved some hypothesis of interest that we've predefined. There are some special design considerations, though, when we're doing both Bayesian methodology, but also more generally adaptive designs. The FDA does require that all trials maintain their frequentist operating characteristics. And so this includes Bayesian trials or trials with adaptive components. So this really is motivated by the idea of achieving a desirable power and maintaining a low type 1 error rate. We don't want to include all of these potentially nifty and interesting elements if we're going to really inflate our type 1 error and mistakenly identify ineffective treatments as effective, nor do we want to design a study still, even with adaptive components that has low power, we might not actually identify meaningful interventions. When we try to evaluate these operating characteristics of power and type 1 error rate or what sample size we might need, in a lot of these situations, they're very complex relationships. So we don't necessarily have a nice simple formula we can sort of plug our assumptions into and come up with an estimate. A lot of these rely on extensive simulation studies to determine the power for a given sample size, or to look at various sample sizes to identify what the type 1 error rate might be for your study. And I will mention, just sort of with any Bayesian approach, prior specification can be quite challenging. There's a lot of considerations and possibilities you can use for how you specify them, what that means for you quantitatively, and for the complexity of your study. And so this is a great opportunity to work with your statistical colleagues to help inform what might be appropriate and what might be best for your given situation. So to sort of lead us to a conclusion, I'd like to give this slightly tongue-in-cheek XKCD comic that's looking at this idea of giving us a very high-level overview of what might be a difference between a frequentist and a Bayesian statistician. And they're looking at answering the question, did the sun just explode? And it's nighttime, so we really can't just look out our windows and tell. So they've gone through all of this work to make a neutrino detector that measures whether the sun has gone nova. And then, because why not, it rolls 2 dice, and if they both come up 6, it lies to you. Otherwise, it'll tell you the truth. So they decide, since it's nighttime, to try this nice invention out. And so they ask the detector, has the sun gone nova? You hear the dice roll, it tells you yes. And so we now have sort of the outcome or conclusion from our neutrino detector. And we can see how our frequentist and Bayesian statistician might respond. Our frequentist statistician says, okay, well, you answered yes, and you could potentially be lying to me. However, the probability of you lying, let's see, 1 dice is a 6, the other dice is a 6. So 6 times 6, okay, 1 out of 36, 0.027, so it's less than 0.05. Ergo, I guess that's pretty rare. So you probably didn't lie to me, so the sun probably did explode. And then the Bayesian statistician, using his prior knowledge, says, bet you $50 it hasn't. So again, a very tongue-in-cheek, I appreciate both methods very much, but I think it's a nice illustration of there's situations where, you know, incorporating prior information maybe is more beneficial than relying on some device with some probability of lying to you to tell you if the sun's exploded. So as a sort of wrap up, and this is also included in a very similar format in your worksheet and workbook, is should I consider an adaptive trial or an adaptive design or adaptive components for my next study? So here I just highlight some of the advantages and challenges of these designs to keep in mind when you're evaluating the appropriateness of these methods. One advantage, and the really big one, is the increased flexibility. You're no longer stuck with maybe what you designed in advance and you can't change it because you didn't plan to change it. You have a lot of flexibility to incorporate uncertainty. There's also the potential for more efficient use of resources, both financial and administrative, depending on if you're doing seamless designs or including multiple treatment arms in a shared control versus a series of standalone trials. There's also the potential, which people generally are attracted to these designs for greater statistical power in the right context and situation. So you can gain some advantages in that respect. There's also the ability to generally answer broader questions with a lot more complexity because we have a lot more moving pieces, which is also potentially a challenge of our study. One of the challenges, though, is that these methods and approaches to design do need advanced analytic methods to help reduce your chance of making those type 1 errors that the FDA does not want you to make. There are gains that you might have in efficiency that naturally result in some trade-off with some other piece of your trial. So even in this case of maybe doing an interim analysis to stop for futility or efficacy, by taking a peek at the data, we're going to need more subjects overall if we have to enroll the entirety of the trial than if we had done just a fixed design with no interim monitoring. There's also just more challenging logistics because, again, there's more moving pieces, and if it's like a master protocol, you have a lot more complexity to address and enumerate and elaborate on in the guidance, the guidelines, and predicting what might happen. And also just generally, you might think an adaptation or an adaptive method might be really ideal for your situation, but it actually might be limited by the question or science at hand and just constraints of reality. Again, if you're looking at some outcome that's 10 years in the future, you're probably not going to have the ability to adaptively randomize based on outcomes in that situation unless you're really, really in it for the long haul for that one study. And so with that, I thank you for your attention for this adaptive trial discussion.
Yeah, if you want to ask a question.
Can you talk a little bit about adaptive modeling, how that can be helpful or not?
Yeah, so the question was talking about adaptive design modeling and how that can be helpful or not. And so Heather, I'm wondering, are you thinking more about like the assumptions you're making for different models and how you evaluate those, the appropriateness maybe thereof or not? Okay, and so in that case, it does really rely a lot on that one bullet point I had on one note about extensive simulation studies. And so the most important thing we do a lot of times when we look at these adaptive trials is we say we're making some assumption about these maybe outcomes and the distribution of those outcomes and what we might expect to see. Now, of course, these are all built on assumptions, and maybe what we hope to see is not actually what we will see. And so in situations there, we evaluate them a lot by doing sensitivity analyses where we assume either worst case or harder scenarios to say, well, what would happen if we had less of a response rate, or we had a bunch of people drop out of the study that we didn't anticipate? Would we still have adequate power, or would we need to change our assumptions, or do we even make the correct assumption for maybe the distribution of an outcome? What if it actually was something else that we misspecified? And so a lot of that comes down to just rigorously evaluating a lot of potential outcomes until you feel confident that either you have a rigorous design that isn't super sensitive to deviations, or you're really confident that what you think you're going to see is, in fact, what you'll see.
I know this is a lead into the next talk, but it seems like platform designs are smart. Is this true?
So the question was, leading into the next talk, kind of, it seems like platform designs might be smart designs. Is this true? There is some potential overlap in the design structures, but I think some of the difference, as I'm sure David will talk about, is the end goal of inference. So a lot of times I know smart trials are looking at these sequential decision strategies as people maybe would go through treatment, whereas a platform trial might be saying, okay, I have like 2 comparisons, and can I drop one to add a totally different arm, just identifying that one early arm maybe is not efficacious and shouldn't be given to people at all. Whereas I think in smart trials, the hope is that all the included therapies might be ethical to give and might be beneficial for the right person. It's just a matter of identifying where their trajectory might optimally go.
choices as you conduct an adaptive design, like particularly if you have, say, 2 or 3 arms in the beginning, and then you drop one of them and you add something else, or one of these complex platform design. I can imagine that some of the study sections may want you to justify that really well. So I wonder if you can comment.
Yeah, so the question was, how does one maybe go about justifying these adaptive designs, especially things like the platform design with the Woodcock and Lavange example, where there's a lot of moving parts, things coming in and going out and changing over time. And how might you justify that to a study section or like just other stakeholders to actually get them to buy into the design? And so I will admit this is a case where there's a lot of complexity I think that people don't value. And so one example is I was recently reviewing a design proposal where they said, I want to do this adaptive platform design where I can add things in and take them out. And they were proposing a grant for 3 years. And I was like, I just don't know if 3 years is long enough. And also, I don't know, you didn't mention like, well, what's the next stage? Like, do you have more funding lined up? Is there like a hard stop at 3 years? I mean, so in situations like that is actually sort of the opposite. I wasn't convinced that they had thought through all the implications of this design type and shown the concept and proof of that. Otherwise, though, if you can show sort of the resource side is maybe met or you have a long-term plan for this in perpetuity design, I think a lot of it comes down to illustrating through rigorous simulation studies. I've looked at a lot of scenarios and a lot of potential cases. And so what happens if actually all of the interventions perform similarly and none of them are actually standing out as beneficial? What do I do then? Like, can I show that I have some strategy in place or some way of addressing that limitation when it's not a clear cut, this is better than that, drop that arm? Again, I think this is one of those cases where there's a lot of hype around adaptive designs, and I think a lot of potential for them, but there does need to be a lot more consideration for the resource constraints and the potential outcomes that might not be as ideal as you might hope in those designs.
And perhaps that's where you need more of the stakeholders or clinicians, some sort of consensus so that you can come up with that strategy for
yeah.
designing.
Yep, and Elizabeth is right that it's really important to have the stakeholder consensus and at least stakeholders involved, not just sort of the statistical features, but like what are the implications of these designs for people involved and for people who might enroll in the study, and what might they benefit from or not get compared to maybe a more traditional study design.
It seems like the time frame is longer for these decisions. And I know a lot of the NIH grants have more time frame. Agencies like ours tend to want to compress it painfully into a very short time frame. But as you do more work in this area, how do you develop that funding and potentially incorporating more elements that go along?
Yeah, so the question was, how might these adaptive designs, since they do seem to need a longer time horizon to successfully implement, how might that alter or affect sort of funding considerations, since some agencies have some longer timeframes, but others do not have that sort of luxury to have some time to work with? And so I think one of the things is that these are newer considerations and more recently have been shown to have some statistical benefits. So I think it's now turning to more of a consideration in the dialogue with stakeholders to say, if we're really interested in this, should we make new mechanisms or new frameworks to fund these sorts of designs? And so again, the 3-year grant for that platform trial I was reviewing, I was like, there's a lot of questions I wish I could ask these people about. But if we had some mechanism, that it was a great motivation and great idea, but just those practical constraints. And so I think some of it's just continuing evolution of funding mechanisms and streams, and people like the FDA and NIH coming to terms with the fact that there are these newer pieces that people might be proposing or incorporating that need either more time or oversight or consideration than we historically have had for the designs.